The overall goal of the following experiment is to ascertain how RNA molecules fold using hydroxyl radical footprinting. This is achieved by end labeling, gel purification, and pre-folding of the RNA, which ensures conformational integrity of the RNA prior to magnesium-mediated folding. As a following step, the Fenton reagents are mixed to generate hydroxyl radicals which can subsequently cleave the RNA backbone according to its solvent accessibility. Next, the RNA cleavage products are separated by denaturing polyacrylamide gel electrophoresis and visualized by autoradiography. The band integrals are quantified by semi-automated footprinting analysis software. The ultimate goal is to generate folding isotherms reflecting tertiary structure formation of RNA. This is accomplished by scaling the normalized band integrals to fractional saturation. The isotherms can subsequently be fitted to the Hill equation.
The main advantage of hydroxyl radical footprinting is that you obtain tertiary structure information of RNA using inexpensive chemicals. Due to their high reactivity and small size, hydroxyl radicals are perfect probes to distinguish between solvent-accessible and buried nucleotides. This method can help answer key questions in the field of RNA structural biology, for example, how do ribozymes employ metal ions to reach their active conformation? Hydroxyl radical footprinting can be used to understand the fundamentals of RNA specificity and recognition. Besides revealing information about RNA tertiary structure, this method can be used to determine the precise protein binding sites in RNA or even DNA molecules.
The major challenges of this method are to prevent sample degradation by ribonucleases and to optimize iron concentration in order to obtain the right amount of RNA cleavage. Also, the detailed analysis of the band intervals can be quite tricky.
The main benefit of this video is to help the audience understand the planning stages and successful execution of hydroxyradical footprinting experiment.
Prior to the start of this protocol, prepare all footprinting reagents as described in the written protocol accompanying this video. RNA can be produced, dephosphorylated, and end-labeled as outlined there. Purify the radioactively labeled RNA using denaturing gel electrophoresis. Recover the purified RNA by 2 subsequent gel extractions using 0.3 molar sodium acetate. Then, precipitate the RNA by mixing 0.5 milliliters of the aqueous solution with 1 milliliter of ethanol. Incubate the mixture for 1 minute on dry ice. After spinning the sample, discard the supernatant. Wash the RNA pellet with 70% ethanol. Remove the supernatant after additional centrifugation and dry the pellet in a vacuum. Next, dissolve the purified P32 labeled RNA samples in 330 microliters of 1x reaction buffer. Pipette 30 microliters of the RNA solution into a new reaction tube. Precipitate the RNA with ethanol and then dry the pellet in a vacuum. Once dried, this pellet can be used for generating the reference ladder as described in the written procedure. Denature the remaining buffered RNA solution by heating at 95 degrees Celsius for 2 minutes. Cool the samples to room temperature for 15 minutes and follow with a quick spin to bring the condensates back into solution. To begin the footprinting experiment, set up 27 reaction tubes for enough samples to fill a 30-well electrophoresis gel after including the ladders and uncleaved control. Determine the final magnesium ion concentrations so that they are evenly spaced on a logarithmic scale over several orders of magnitude around the titration midpoint. Preparation of the magnesium ions in 1x reaction buffer is described in the text. Separately incubate the RNA and the magnesium ion solutions at 50 degrees Celsius for 5 minutes. Then, mix 10 microliters of the RNA solution with 90 microliters of the corresponding amount of magnesium ion solution to reach a final volume of 100 microliters. Incubate each mixture for 30 minutes at 50 degrees Celsius. Subsequently, allow the solutions to equilibrate at 25 degrees Celsius for 1 hour while RNA folding occurs. Meanwhile, immediately before initiating the hydroxyl radical footprinting reaction, prepare the Fenton reaction mix as described in the written protocol. Prepare the peroxidative reaction by pipetting 2 microliter droplets each of iron EDTA, sodium ascorbate, and hydrogen peroxide at the top inside of the reaction tube containing the RNA solution so that the droplets are not touching. Start the footprinting reaction by vigorous mixing. After 15 seconds, stop the reaction by adding 300 microliters of pure cold ethanol. Turn the tubes 3 to 5 times. Finally, precipitate, wash, and dry the pellet as performed earlier. As an alternative to the peroxidative reaction, the oxidative hydroxyl reaction can be performed by adding 5 microliters of the freshly prepared Fenton reaction mix to the samples. Incubate the oxidative reaction for 30 minutes at 25 degrees Celsius. Then add 300 microliters of pure cold ethanol to quench the reaction and mix by turning the tubes. Once again, precipitate, wash, and dry the pellet. Upon completion of either the peroxidative or oxidative reaction, dissolve the dried RNA pellets in 8 microliters of gel loading dye 2. Confirm that the P32 labelled RNA is resuspended by using a Geiger counter. Prepare a denaturing 8% polyacrylamide sequencing gel according to standard protocols using a 30-well comb. Load the samples, including 2 references and an uncleaved control. Separate the RNA fragments at 60 to 75 watts for 2.5 hours. Expose the dried gel to a storage phosphor screen overnight. Scan the phosphor screen with an imaging system for filmless autoradiography. Then, transfer the gel image file to the computer for analysis. To begin data analysis, open Sapphire, an open-source software for single band fitting and quantification. Load the RNA sequence as a .txt file. Followed by the gel picture as a .gel file. Define the lanes and adjust the band intensities. Then choose an anchor lane and perform a gel alignment. Assignment of bands to nucleotides occurs in reference to the RNA's T1 digestion ladder. Quantify the band intensities and use the normalization/color plot feature to normalize and assign potentially invariant residues. Save the output as a .txt file. Sapphire outputs a spreadsheet containing columns representing lanes on the gel and rows representing the individual band integrals corresponding to the RNA fragment. First, the protection sites that display a noticeable change in solvent accessibility have to be identified by comparing the protection profile derived from the no magnesium ion sample to the profile of the endpoint magnesium ion concentration. The lower the value, the more protected the nucleotide is against attack from the hydroxyl radicals and vice versa. Next, create transition curves of band intensities of individual or groups of nucleotides versus magnesium ion concentration using a spreadsheet format. Individually scale these transitions to the fractional saturation function as described in the text portion of this protocol. As a final step, fit the data to the Hill equation. This analysis scales the transition to fractional saturation, determines the transition midpoint, and provides a phenomenological test of whether a transition is sigmoidal. Shown here are representative results from P4-P6 RNA hydroxyl radical footprinting experiments. Isotherms were derived from sequencing gels, analyzed by SAFA, and then fit as described in the analysis section. The gel image indicates that background cleavage is minimal, and that single nucleotide assignment is possible due to well-defined bands in the T1 lane. The hydroxyl radical-induced fragmentation of RNA is well above background. The transition from low to high magnesium ions is selectively affiliated with decreasing intensity of individual and groups of bands indicating formation of RNA tertiary structure. Protection refers to single or groups of contiguous nucleotide whose cleavage changes concomitantly. Band intensities are quantified and normalized by SAFA analysis. The output is a thermal plot visualizing the degree of protection against hydroxyl radicals. The color transition describes the accessibility change upon magnesium ion addition. White to red shows more accessible nucleotides, whereas white to blue shows more protected nucleotides. Each degree of shading is affiliated with a numerical value which can be plotted as a protection curve and analyzed by a binding model such as the Hill equation. In this example, the equilibrium dissociation constant affiliated with protection 153 through 155 is roughly twice that of the corresponding value of protection 163 through 164.
This hydroxyl radical footprinting experiment can be done in 1.5 days if it's performed properly.
While attempting this procedure, it's important to remember to wear gloves and a lab coat to avoid any RNase contamination. Also, the final iron concentration depends on the experimental system. Therefore, we recommend performing a dose-response experiment before footprinting.
Following this procedure, other methods such as time-resolved hydroxyl radical footprinting can be performed in order to answer additional questions. For example, at what critical time point do RNA molecules reach a misfolded or active conformation?
After its development, this technique paved the way for researchers in the field of structural biology to explore inter- as well as intramolecular tertiary interactions of nucleic acids.
After watching this video, you should have a good understanding of how to determine tertiary structure formation of RNA molecules. Don't forget that cacodylates and phosphorus 32 are hazardous. Precautions such as wearing gloves and protection goggles, as well as the use of plexiglass shielding, are recommended while performing this procedure.